Good afternoon, everybody. Actually, it's so wonderful to finally have a partial face-to-face meeting. So many of us are here, and some of us are still on Zoom. So this is the classical hybrid meeting. Welcome, everybody. We are extremely honored today to host Professor Musa Yudim, who has just recently received the Israel Prize for Science. That he absolutely deserved for a career, time, contribution to the neuropharmacology of Parkinson's disease. Musa's initial steps, he just told me, started in 1965 when he first touched the MAO-B inhibitors, and since then, and throughout his career, he has focused on bringing those two molecules into clinical practice, and it really changed the way we treat Parkinson's disease these days. Initially with selegiline and then with rasagiline, which is now a drug that we all give to our patients on a daily basis. This is the first drug we would give. Musa has done much more than just developing drugs and bringing them into the clinical world. He has really studied and explored and contributed significantly to understanding the pathophysiology and the neurochemistry of Parkinson's disease. As a professor in the Technion throughout his career, now he's the emeritus of the Technion. He's also involved in developing new drugs that we will hear today. Can I mention your young age? At the age of 82, he's still active in bringing new ideas, new combinations of drugs, and his dream is to have the new levodopa that will have no side effects, that actually will cure the motor Parkinson. And we will hear about it more. So thank you, Musa, for taking this opportunity and sharing with us your view and perception on Parkinson's and whatever it is.
I'm really honored to have received. I've received many prizes, but to receive the Israel Prize is a big trouble. I told Amos, I'm not a real scientist because I got Israel Prize. I haven't been giving a talk for a long time, and so today I'm going to tell you, I hope, something new about the new approach to how does this move. Yeah. So we know that Parkinson's disease is more complex than when I started way back in 1973. And we want actually today together with Peter Ritter and Berkmeier, we published one of the first papers that Parkinson's patients suffer from depression. And it wasn't that they found out about Parkinson's disease, but we know now that they have predisposition to depression. We know that the serotonergic and noradrenergic neuron in the rapidly degenerate, source of the midbrain dopamine, and eventually frontal cortex cholinergic system with dementia. So it's a complex disorder. And yet we are being foolish trying to treat this drug with one drug, mainly dopaminergic drugs. Oh, do I have to press that to change?
The arrow.
Then I lost. So these are some of the drugs that are available, you all know, and they're used in the clinic. None of them really show the disease modifying. There was some suggestion that rasagiline, the drug that we developed, could be. There's still some people still believe that it might have a disease modifying, and they suggest that rasagiline should be used before giving L-DOPA, at least 1 or 2 years before. And so, but it's still not a drug that we think is really. Now, the whole idea is whether we can find a drug that can be neuroprotective once you have the disease, so the disease remains constant, or if we have a drug that causes neurorestoration. And today I'll tell you about one or two drugs that we've developed that actually causes restoration in the dopamine neurons. And we're very excited about, and we discovered this by accident. Everything happens to me by accident, which is good. That's the beauty of pharmacology, serendipity. We know that Parkinson's disease and Alzheimer's disease, all the neurodegenerative diseases, show this complex pattern of what may cause the diseases. Actually, when I was an NIH or a faculty scholar in residence at NIH, I was 1 year, I read about cell death in all the diseases, liver cholestatic disease, nephrotoxicity. Everybody saying the same thing, the whole thing, but nobody knows how the cells actually die. That's wonderful. But we know they all converge to mitochondria. And in 1962, when I started working for my master's degree, I worked on mitochondria because monominoxidase in the mitochondria. In those days, everybody was interested in the electron transfer system in mitochondria. Nobody cared about anything else mitochondria might do. But later on, of course, when apoptosis became, we found that mitochondria is responsible for cell death, cell survival. And so even in cancer, they're going towards mitochondria. Everything actually we're doing in neurodegenerative diseases is completely opposite to what they do in cancer, the same mechanical system. So one of the things we discovered is iron accumulation that we found in every neurodegenerative disease that you can think of. Where the neurons die, iron has accumulated. I published it in Encyclopedia of Neuroscience, the whole article about it. And every animal model, you see the same thing. These are the animal models. This is 6-hydroxydopamine, iron accumulates when you degenerating, iron accumulates in the compactor. This is the spine in the ALS model, in the spinal cord iron. This is lactocystin, one of the models that we developed for Parkinson's disease. We put it in inhibitor. As you can see, iron increase, and this is with kinase, and so possibly an Alzheimer's model, iron accumulation. So the question, why is this iron always appearing in every neurodegenerative diseases when the neurons die? So, and we know that iron plays an important role in the oxidative stress. We know that iron increases in brains of Parkinson, no question. In the compactor, not the reticulator, and within the melanin-containing neurons. Very important. So we came up with this concept that has really stood the test of time. Everybody uses this to describe their own oxidative stress in neurodegenerative disease. We know that iron accumulates. We know monominoxidase increases because of glyosis, so more hydrogen peroxide is produced. So penton reaction leads to the oxidative stress and maybe eventually results in cell death, because we don't believe apoptosis plays a role. We don't know how the neurons die, but maybe contributes to oxidative stress to cell death. So we decided that we were going to develop not a single drug, but a multi-target drug, like an octopus with the little tentacles. The idea was to take one drug and add to it different active moieties, like monominoxidase inhibitor, cholinesterase inhibitor moiety, and iron chelating, and anti-reactive. So this is our concept of multi-target drug that we developed years ago. And we started to do this because we discovered that rasagiline, this moiety of rasagiline is really important, the propargyl moiety. The propargyl moiety does everything that the Steve Jobs, it was a drug that I co-developed with Martha Weinstock, and I talk a lot about M30 or iron chelated, but this propargyl moiety really is involved in cell survival. It's the neuroprotective moiety of rasagiline. We've done all the molecular mechanism, and all of this activity results in this little molecule. So we decided we take this molecule and use it to put into different molecules, drugs. So that's the propargyl moiety is a crucial organ. So we made a number of drugs. So first, sertraline, the first drug I discovered with Peter Ritter. And so this is very sticky. So we took the propargyl, we took the cholinesterase inhibitor of radistignine and put it into rasagiline with larestigium. And then larestigium failed because Martha used a lower concentration, and later always drugs fail at the dosage level. And so here the lower dose of phase 2 it was negative because she's the lower concentrator. That's not important anyway. But we had developed an ion chelator called VK28 years ago, together with Avram Wachowski and my brilliant Chinese student, Hailing Zhang. And we decided we modify this and we put this, we made different derivatives of VK28 and we put propargyl moiety into it. So I will be talking a lot about this compound M30, that's a propargyl, and this compound that has a cholinesterase, pseudocholinesterase inhibitor carbamate. So we have a really like an octopus, different moieties. And we know that these drugs inhibit MAO and they come off, and the pseudocholinesterase inhibitor also inhibits choline and comes on. So you're left with a basic ion chelator at the end of the day. I won't go through in all detail. We know that M30 is a brain selective MAO and B inhibitor and not the liver and small intestine. It increases the brain level of neurotransmitter, noradrenaline, serotonin, and dopamine because it's an MAO-MB inhibitor. It has an antidepressant activity, no question. In a post-swim test, it's classical antidepressant. It shows the same thing because all MAO-A inhibitors are antidepressant, and our drug is MAO-A as well as MAO-B, irreversible inhibitor. It has antidepressant activity, and it doesn't give you the cheese effect. We can discuss the cheese effect later on because it's one of the side effects of some MAO inhibitors. And so what is happening to M30, our compound, what is M30P, which has the propargyl as well as the cholinesterase? It breaks down to M30, first of all, then the propargyl moiety comes off because it attaches the APPD of the enzyme, and you're left with the ion chelator that eventually gets metabolized again by MAO. It's an AMI, and you get again hydroquinone, which is also a chelator. So we have a drug which, when it metabolized, is also an ion chelator, antioxidant. So we have something, I think, quite unique in the drug. Of course, it prevents MPTP neurotoxicity because of the B inhibition. But I want to go really on neuroresearch, and this is really the most exciting part of our book. One of the first things that we discovered was that to take our ion chelators and incubate them in absence of serum. Here we use the SSY cells, and I can see that it causes these cells to differentiate, and gap 43 goes up dramatically, suggesting the drug has neurotrophic activity. No question. We've done it in NT3 cells, we've done it also in hippocampal cells, many. The drug has neurotrophic. We said, if it's neurotrophic activity, can we see the neurotrophic activity in vivo model of Parkinson's disease? Lesion the dopamine neurons, give the drug, and see whether we can restore the neurons. First of all, you put it on embryonic stem cells. You can see they all converted into different neurons. There was dopamine there, glutamate in there. We did studies in the United States with colleagues. So our first study was using as you know, ubiquitin has been implicated in Parkinson's disease and Alzheimer. And the ubiquitin, like the cysteine, is an inhibitor of UPS. So together with colleagues, we decided to inject ubiquitin. First of all, we give like the cysteine unilaterally because it doesn't cross the blood barrier. You can see that dopamine neuron in the compactor degenerate after 24 days. So we gave our first ion chelator, and we saw it protected completely, and M30 completely protected against lactocystine. That's neuroprotection. Okay, that's okay, but unfortunately, we don't have a biomarker for Parkinson. Who's going to get Parkinson's disease? If we have a biomarker, we can identify that the patient is going to get the disease in 10 years' time. I believe if we give a neuroprotective drug, we could prevent the onset. But it's too late. By the time we discover the disease, it's too late. You're falling into the Grand Canyon. But this is interesting. After we lesioned the thing, we gave the drug, VK28 and M30, and the neurons came back. No question. We've done the same thing in the MPTP model. Lesion in the MPTP model. Here is in the MPTP model. And in the MPTP also, giving the drug post-MPTP, 20 days, 22 days after MPTP, the neurons come back. I've got even more beautiful. And we've done it in the sick called dopamine, same thing. We had some data, I think rotenone, I don't have it in the rotenone model. There's no question that when you give this drug post neurotoxin, it causes the neurons come back. And in a study that we did with Julian Nunes in Chile, you can see that it causes dendrite formation. This is with dextroxamine, which is the classical iron chelator. Unfortunately, it doesn't cross the blood-brain barrier like our compound. And this is with our, again, sorry, I think I missed it. I should have the monominoxidase. You can see that here with M30, it causes restoration of type G with M30 when you give so we are very excited that you can see with MPP, it completely protects and dendrite formation is restored. So the drug has something unusual. Well, and of course, we are interested to see what is the mechanism. This is another one, this is mesenteric neuron, and you can see that it stores the functional dendrite. And when you merge it completely, you can see. And the question, what's the molecular mechanism that this drug has such a wonderful neurorestorative activity? This is from another MPTP model that we did, and you can see that here is cell line, here is M30 itself. We give MPTP, all the neurons are lesion, and when we give the M30 after MPTP, the neurons come back. There's no question the neurons come back, and of course, the number of cells and tyrosine hydroxylase activity is restored. And this is another one with GRK2, which is a G protein regulated input rectified potassium channel that is found in the nigra. And then you can see that when you give the MPTP, it disappears, and you can protect post MPTP. You can restore this protein. And we also went back to neurogenesis. We thought maybe this drug really have neurogenic activity, and so we did some VRDO analysis, which I'm one of my students, which we published later on. And what we could not see any VRDO staining in the substantia nigra, but we found it in the hippocampus. I think people have found neurogenesis in the hippocampus, in the dentate, but we could not find it in the nigra, although there are controversy in the literature whether neurogenesis can occur in the striatum at least. But what we couldn't find, as I said, we found out that there was neurogenic activity, so this thing there must be a special mechanism for this. So I want to go back now to try to find out why M30 and the other chelators that we've developed have this unique property. As you know, my group with Peter Ritter and Gideon Rechavi were the first in the world to use transcromatomy to look at what happens in Parkinsonian brain in the compactor when it's generating gene analysis. We use a gene chip of 27,000 genes. Our controls were substantia nigra compactor, reticulata from Parkinson, reticulata and compactor from Alzheimer's brains, hippocampus. Anyway, we had a very good control. Anyway, we discovered, yes, in the nigra of Parkinsonian brains, which were obtained thankfully from Peter Ritter, 4 hours after that, we could get brains, isolate the RNA, and do the analysis for the gene analysis. And these are the unique genes that we discovered. I won't go through all of them. As you can see, they've been implicated in. But one of the things we discovered, which was after we published, we didn't know, was this gene called EGNN. The expression of this gene is dramatically increased. We didn't know what this gene was. We went to the literature, we discovered, yes. This denotes the enzyme proline 4 hydroxylase, which is an iron dependent. In fact, proline hydroxylase is the iron oxygen sensor of the cell. Tremendous area of research which you didn't know, and the expression of this thing is up. When the activity of EGNN goes up, which we have seen, by the way, in Parkinson's brain, angiogenesis, cell proliferation, iron metabolism VGF and many neurotrophins go down. When the activity of this enzyme goes up, these go down. Okay. And this enzyme is an iron chelator. There was a paper in JBC that related if you had an inhibitor of proline hydroxylase, you could reverse this and you get angiogenesis and you get the neurotrophins going up and the HIF, which is very much hypoxia-inducing factor, will go up and will be very important. So, just to go back, a number of drugs are now being developed for Parkinson's disease immunotherapy. I think they're going to fail exactly how they failed in the beta amyloid for Alzheimer's disease. I don't believe it. And anyway, alpha-synuclein and amyloid precursor are iron-regulated proteins. They have within their mRNA 5-UTR exactly like ferritin. They are iron-regulated proteins. So iron chelated do regulate alpha-synuclein. And I'll show you one example that we did. Here's a study that we did in MPTP. As you give alpha-synuclein, it goes up. You give M30, 5 milligram, it goes down, 10 milligram, completely disappears. There's no question that iron chelated regulate alpha-synuclein. So immunotherapy, I'm not sure in chapter immunotherapy is going to be important for the treatment of Parkinson's because it's such a complex disease. So EGLN, I mentioned, is a proline hydroxylase iron oxygen sensor. When it goes up, you get iron-induced oxygen of HIF, and all of these things go down. And I mentioned to you that the paper in JBC suggested that if we had an inhibitor of EGLN, we could cause these neurotrophins to increase in the brain and maybe affect cell cycle. So we published the paper about 2 years ago, that yes, proline hydroxylase is increased, not only in gene expression, but also is increased in the brains of Parkinsonian patients. And we saw that our compound actually inhibit nicely proline 4 hydroxylase. Very nice. So this was really the beginning of saying that maybe the drug is working via HIF. And indeed, we saw that if we inhibit with the different iron chelators in cell cycle, in the NC34 cells, we get significant increase of HIF, hypoxia-inducing factor within these cells. We've seen, and as a concern, we get VEGF, enolase, and BDNF also increasing in these cells. So this is this the way the drug is really working? And by the way, the drug is permeable in the brain, unlike the spiroxamine. So, you know, if you are able to increase BDNF and VEGF and GDNF by a small molecule that crosses the blood-brain barrier, I think it's important because, you know, the studies that were done clinically with GDNF, did they do the BDNF? I think GDNF, injected GDNF in the brain. It didn't work because a big molecule doesn't cross the cell membrane. I don't know what they did, but our drugs actually increase. I'll show you later on that it increases all these neurotrophins in the brain. Here you can see that BDNF and GDNF is published in neuroscience that yes, they increases the level of the neurotrophin factors in the different brain areas, and of course, hypoxia-inducing factor is also significantly increased. What is all this got to do with it? Because we know one of the things about hypoxia-inducing factor is a big literature that it can regulate cell cycle. Cell cycle is a very complex. I wouldn't say I'm an expert. We know that there are different stages of cell cycle. Cell cycle can also cause cell proliferation, which becomes cancer, or cell differentiation, making this differentiation in cell, etc. So we know that cell differentiation and cell survival is one of the aspects of hypoxia-inducing factor. And we saw that indeed, we can see that the hypoxia-inducing factor is significantly increased by our multi-target iron chelators. And so the question is, does our hypoxia-inducing factor, the iron chelated, cause this cell arrest and cell differentiation? Because we know that G0, G1 is an important step in cell differentiation. If it goes to G2, it goes to gel proliferation, and that's not good. That's not good for us. And so we did, and we can see that cyclin D significantly decreased. So this is yes, that maybe the drug is really causing initiation of G0, G1. That means the cell is going to cell differentiation rather than cell proliferation, which is one aspect of the drug. And we did, of course, fact studies, and we showed yes, G1, G0 is increased, and G2 is decreased. So this means that we're on the right track. The drug is really affecting cell cycle, and maybe cell cycle is responsible for these neurons coming back in the models of Parkinson's disease, and also differentiation that we see in cell culture. But we still haven't answered the question of mitochondria, because we know that mitochondria is responsible for cell survival, cell death. We know that in Parkinson's disease and Alzheimer's disease, as well as ALS, there is a defect in the mitochondria. There's no question. Probably in all neurodegenerative diseases, there's a defect. And the study that we did is an incredible paper that I think one of the most quoted papers now in Science and Medicine, in which we studied 480 Parkinsonian brain analyzed gene analysis, and we came to the conclusion that activation of PGC alpha, which is the one really starts activating mitochondrial synthesis, is important, could be an approach to treatment of Parkinson's. If we have a drug that can activate PGC alpha, it will be an important drug for Parkinson, because it's really the mitochondria which is really a crucial aspect of the survival of the neuron. So this is what really happens is that acetyl PGC alpha is deacylated to PGC alpha, and then the PG alpha acts within the nucleus for the transcription of many of the components of mitochondria, and then eventually the nuclear transcription factor, and then it gets the nuclear activates the mitochondrial factor. And this was important. Does our drug really, because we saw this cell cycle, it must be somehow related to activating the mitochondrial system. And so we've done a lot of studies on this compound. We measured SIRT is significantly increased at extremely low concentration. Too much of a drug is no good. Every drug has a biphasic effect, even propranolol, which is used clinically. And we see that, and one of the discoveries of rasagiline and salicylic that has neuroprotective activity in vitro in cell culture, etc., was that at a lower concentration they have neuroprotective activity, and only MAO inhibition is at a higher concentration. And so we've seen that yes, SIRT is significantly changed in response to kainate and glutamate. It protects. Again, and you see PGC alpha is being increased also in response to glutamate and kainate in the hippocortical and hippocampal culture. As you can see, it's significantly increased. And again, here is this tunate kainate, again, same thing, PGC alpha mRNA is significantly increased via the two compounds. I here only give you the HLA20 rather than M30. We got a similar thing with the M30. I think most iron chelators will do that, because all the iron chelators that we have examined have this neurorestrictive activity in the models that we have used so far. So finally, we see that TFAM, the transcription factor, is also increased via our drug HLA20. So in one summary that I want to put you in is that we believe that iron chelators activate SIRT, which is PGC alpha, and PGC alpha affects NTFS, and then transcription factor mitochondrial factor. That's one approach. We also recently found papers showing that actually PGC alpha activates hypoxia-inducing factor the same way as HLA-20 and 30 that inhibit proline hydroxyl and causes hypo. So there is a combination of activities going on and the increase of the neurotrophic factors, which I showed you, suggesting that the neuroprotection, neurorestoration, and neuroplasticity may result as a consequence of two pathways, the activation of PGC alpha and inhibition of proline hydroxyl, which results in hypoxia-inducing factor increase. So, oh, this is the wrong one. Sorry. What we believe is now what M30 does is really a complex mechanism of action. First of all, I think I'm going to give a talk next week or 2 weeks' time. And as I mentioned, we've done a lot of work with this compound, tremendous amount of work in the Alzheimer models that regulates amyloid precursor protein because it's an iron-regulated protein. I mentioned to you that the PKC pathway and etc. But the important thing is that the drug really is inhibiting the thereby pool of iron, which is important, and this the increase in the neurotrophic factors. We believe this, and which really relies on changing the cell cycle that I mentioned to the G0, G1, and the increase of the neurotrophic factor, especially GDNF and BDNF and erythropoietin. Finally, one last drug that I've developed recently. All the cholinesterase inhibitors that are on the market or are in the clinic do not have any neuroprotective activity like rasagiline or selegiline. I think the field of neuroprotection really started with the drug selegiline and rasagiline, that they do have neuroprotective activity in the animal models and in cell culture. So what I did was we wanted to develop a new drug for Alzheimer and Parkinson's disease, because we know that Parkinson's subjects suffer from cognition. Rivastigmine has been approved for treatment of cognition in Parkinson's patients. So what I did was to take that beautiful complex propargyl moiety and put it into rivastigmine. And we have a drug called mp31. We have US patent, European patent, Israeli patent, Japanese patent, which is unique. And we discovered this drug is amazing. First of all, it's an MAOA inhibitor. We know that Parkinson's patients suffer from depression, so do the Alzheimer's subject. And we have that it has antidepressant activity. We know that it inhibits both acetylcholine and butyrylcholine stress, and we saw in Morris waterways that yes, that's cognitive enhancing. And we saw that it regulates these neurotrophic genes as well as the antioxidant effect. So we think this is a unique drug that could be used in the treatment of cognition and the depression in Parkinson's patients and Alzheimer's patients. That's all really the latest drug that we're trying to really further develop, which is under development in our company. So I hope. One more slide. So finally, this really is the way M30 works. It's an area of A and B inhibitor, so it's anti-Parkinson, it's antidepressant. I mentioned that it's iron chelated, so it inhibits a phantom reaction. It inhibits proline hydroxyl because it's iron dependent and it's up. I didn't talk so much about the glutamate. We have a lot of work on that, and we have a lot of work on the regulation of amyloid precursor proteins and etc. So eventually induction of neurotrophic activity, because it increases these neurotrophics, and the drug is brain permeable. That's the beauty of the drug, unlike this desferrioxamine, which was used, I think, probably even in Alzheimer's was used for. So finally, I think the approach for treatment of Parkinson's disease, I think we need either multi-target drug like this or polypharmacology with novel new drugs that can act at the different sites in the brain and at a different site. And I think when I started this, I learned this concept from my children who they were making Lego. They put in all sorts of pieces, Legos, making different. I said, oh, why can't we do this to molecules so we can put different things into most of the people who've been trying to develop multi-target drugs have been taking two drugs and joining them, but they break to the original drug. The beauty of this thing that these molecules get released and get attached to the enzyme, so you have less metabolite. You have less metabolite interaction or metabolite parent interaction. So this is a concept that many people are following our step. There are many new papers now that people are doing a similar thing using small molecules, small moieties to attach to parent compounds so that we have a multi-target drug. And really, I owe much to my colleagues who were my two centers of excellence. Unfortunately, technically retired us, or we continue the work. Especially, I want to mention Sylvia, Tamar, Orly One, and Silva, and Sumit, who did all the work on the neurorestoration with the molecule. Thank you very much.
Thank you very much, Musa. Really fascinating and challenging. I will start first. I think Parkinson is one name for many different diseases, and patients are very different. Do you think one drug for all patients, or how, with your perception over the years, we can target the right drug to the right patient?
Yeah, that's a challenge that I even learned when I was at McGill University, or I was in the psychiatry department also. There was the same problem there. How you treat this depressed patient? Do you give an MAO inhibitor? Do you give tricyclic? Because we have no way of identifying which patient reacts to which drug. Is hit and run, I think, normally. And also with the Parkinsonian patient, is hit and run normally. Sometimes they respond very well to sertraline or rasagiline. Sometimes they don't. They respond to L-DOPA or a combination. It's because we really do not, as you said very well, it's a complex disorder, and I think it's different in different patient subjects. So we need a biomarker. There's no question we need a biomarker for the patient. Unfortunately, all these transcriptomic studies have not really shown a true biomarker either for Parkinson's, for ALS, or for Alzheimer's. I think we still need a lot more work with us. I should say that when I showed you that gene analysis that we did on the brain with Martin Rabbi, unfortunately Martin passed away, we thought we really discovered a biomarker of 5 genes that we could identify, by the way, in the blood. We did it. We have still a lot of data on it. And we collaborated with Michael J. Fox Foundation. They gave us a major grant, and they sent us, I think, 1,200 mRNA samples to examine our genes in this mRNA. And we did a study, was double blind. We sent the result back to Michael J. Fox, and they reported back to us that we have discovered with our gene the progression of the disease. The expression of these genes went up with the progression of the disease. We want to work backwards now to symptomatic, etc. We're still doing some work with our gene analysis to see whether we can really identify patients, normal patients, to do as many normal patients who will show the expression of the gene that they eventually may develop the disease. I think that's a long-term study, because we have to wait for some years to see whether these patients who show this expression of these 5 genes will eventually develop Parkinson's disease. It's a very difficult situation. Please.
I have a question. You show not only local patients, but actually neural proliferation. So you have proliferation. With the problem, the cells that proliferate are those that were going to die, or these are cells that were not affected yet by pathology. They proliferate, while others, maybe that already accumulate enough synuclein and God knows what, they are dying. So you have proliferation.
And I call it neurorestoration because there is a controversy, at least in the model of MPD, whether the neurons have really died or they are sick. I don't know what you think of it. So Are the 8 MPT the best model? Maybe cyclohydroxydopamine probably better because I think cyclohydroxydopamine really kills the neuron. Stop, it doesn't. But there is a controversy that maybe the neurons are sick and the neuron, we have lost the tyrosine hydroxide, but you can revive them.
Yeah, that's the question. Do you revive research or cells that have not been affected yet? And maybe
It's a very good question.
and maybe it's good that you still will have enough cells that will make thousand hydroxylates. In parallel, cells that have already made enough cyanogen aggregates die well. It's not that it's not good, but the question is
I
whether
think the models are limited models. They really do not reflect Parkinson's disease. It's even more difficult in the Alzheimer models. We recently been working with Claudio Coelho, who has a rat transgenic model. We've already published one paper with Claudio with our compound, and it seems that the compound really works in his model, which he thinks is much closer to the human. We do not have better models of Parkinson's disease. Even the alpha-synuclein model, which has been used, doesn't fit in. As I mentioned to you, I still believe in this iron because this iron-regulated protein alphas, people forget. People in the field, the industry forget that alpha-synuclein and beta-amyloid are iron-regulated proteins. They forget it. And it's ridiculous trying to make antibody to them to treat the patient with antibody. We still need drugs. Sometimes I give a drug that says empathy is better than stem cell because the stem cell theory also, nothing came out of stem cells really. This is better than stem cells. It's causing neurorestoration in model and causes neuroprotection in the models that we have developed. This was really by accident, which I call serendipity. That's one of the beauty of pharmacology. I want to come back to the cheese effect. This is really a very, very important part of my life and the people who were. Monominoxidase inhibitors were discovered by accident. They were anti-tuberculosis subjects. They were treating them in Chicago with isoniazid and micronizid. And at night, these patients were climbing up, or they were euphoric. They didn't know what was happening. They called the famous Nathan Klein, who became world famous psychiatrist. He said, this drug is acting on the brain. And there's a very famous biochemist in Chicago University called Albert Zeller, will give the result. He discovered that this drug inhibited monominoxidase. It was the first discovery, and it became the first antidepressant. Ibronized. Hoffman and Roche became the most famous company in the world because they discovered the first antidepressant, first brain drug. After 2 years of clinical studies, some patients died, some people developed tremendous hypertensive crisis, and the drug became dangerous, so they removed it. They didn't know what was happening. Until a nice Jewish pharmacologist in England decided to study the files of these patients. They were all eating cheese. Now, cheese is a fermented food. In it, there is tyrosine. You eat tyrosine, is it decarbonated to tyramine? Tyramine is a sympathomimetic amine that is metabolized by monominoxidase. You don't metabolize it, it gets into the cardiovascular system, releases noradrenaline. That's known as the cheese effect. The beauty of selexilin and rasagiline, they inhibit MAO-B. The cheese effect is associated with MAO-A because MAO-A is in the adrenergic neurons in the cardiovascular. So if you inhibit it, you eat cheese or wine. To be bloody-minded, the French call it the wine effect. In American, they call it the beer effect. So it's because they're all fermented food. So when you give them MAO-A inhibitors, you have to tell them you don't eat fish, you don't eat cheese, you don't eat chocolate, you don't eat, the whole list of food you have to avoid. So that's really. So the beauty of our compound, the M30 and rasagiline, and also ladostigine, which we developed with Martha Weinstock, it was by accident. We discovered it only inhibits the brain enzyme and not the systemic, the liver and small intestine. And part of the study that we did showed that the metabolism of this drug is different in the brain with P40, cytochrome P40 versus in the liver. So what we think is happening, why is it inhibiting the brain and not the liver? We think it gets metabolized much faster in the liver before it can. in the brain before it can, in the liver before it can inhibit the liver enzyme. And it's slow in the brain, so it inhibits the MAO. So this is the differentiation between the drug. Again, that was by serendipity that we discovered these action of this multi-target approaches. And I think it's beginning to, you see, in other diseases, they're talking about multi-target drugs and polypharmacology, proper pharmacology, new novel drugs that can act at different sites in the cell that can prevent the cell from dying.
Thank you very much. You have shown the effect of M30 in different models. Would all these models respond to the same concentration? If the same concentration would affect all so different.
We used the concentration that we thought was active, 5 milligram or 10 milligram in all our studies, and it works. I mean, and by the way, other people have studied. We sent the compound to other people and they've studied. No question about this. I know this is a question being asked whether the MAO inhibition and the cholinesterase inhibition, and it does really occur. It's no question about it. I mean, we have got the data shows that. I think it's because I cannot explain to you why we're getting such good results with these the two doses that we've used. Of course, we had other doses. At the lower doses, it's not as effective like at 2.5 milligrams. But it may be that in many would be different. I'm not sure. But in all our studies, I think it seemed that it inhibited the MAO beautifully, inhibited cholinesterase at this concentration. I've been asked this many, many times, but really I have no explanation how he does it. I think it's because it releases the moiety. The most important is that the moieties are released. It's not that the drug is acting on the site. It's not that. It's releasing the drug, it's releasing itself and it's becoming anchelated. So the MAO inhibition, I told you, the proportional moiety goes up, the carbamate cholinesterase goes up, so it goes attacking different sites. That's my only answer to you.
Thank you.
How does it interact with alpha-synuclein? Have you tested if it models of alpha-synuclein deposit?
No, I did it in MPTP. You saw that when we gave it in MPTP, it was up and it really reduced it because it is a chelator. Yeah, I showed you the slide on the MPTP model. When you give MPTP, alpha-synuclein goes up dramatically.
Okay. And
then when you give the M30 before, it really protects it. It's iron-regulated protein, so you prevent it occurring. And we showed that it's got this 5 UTR, like ferritin and amyloid. So I think that's an important aspect.
And the alpha-synuclein comes from this water.
Yeah, you find Lewy body in the colon.
So that's a very good biomarker.
I know you have to ask my neurology friend.
That is the question: how important is crossing the blood-brain barrier, or maybe has to work first peripherally?
That's a very good question. But in the animal models, we see that it's working on the brain.
That's the problem with animal models; they're not really reflecting the clinical part.
Well, you got two successful drugs from working on animal models. All the drugs that we've developed has been on animal models of cell cultures. So
it looks like your drug is acting quite similar to physical activity.
To what?
To physical activity.
I do 8 kilometers every day. I get up and walk every day.
The basic mechanism of exercise is in a very similar pathway as you have shown.
Yeah. It's very important. And one thing is really true. Too much is not being better. Even exercise has now been shown that the exercise also has a biphasic effect. There is not a maximum, the optimum. You can walk, which is beneficial. If you go too fast, it's not as good. You go too really. It's in the May. I saw many papers now have been showing this.
So I have another question about females and males. Because I heard that Parkinson is more prevalent in males.
Do you see any difference? I mean, do you
I didn't hear. Take that thing off.
I'm going to content. So if you test, have you tested the drug with female mice or female rat
Gender.
and gender?
Is it gender related?
Is that because it's more prevalent in males? So that the Parkinson is
I
more.
don't think it would make a difference.
I don't. So why is it more prevalent in males?
Is there?
Yeah, Parkinson's is more prevalent
In man?
in
Yeah. I don't know, maybe it's because the females, you know, they have,
Stronger.
well, who's very
So
interested.
they can get Alzheimer's. So you say they're stronger.
Maybe it's something to do with the when I was no, it's really
The
it could
clinical course is
it could
also
be very-
wider in women.
it could be very important.
And here the division.
Do you think people have more estrogen than men? I tell you why. When I was at Merton Center in Queen Charlotte Maternity Hospital, we were studying the cycle of older women, you know, menstrual cycle, and we were getting endometrium from them, and we were looking at monominoxidase activity. Well, this we discovered, yes, the monominoxidase activity in the endometrium changed with the cycle because you get changes in progesterone and estradiol. That's how we discovered that progesterone, and this was at the time with the progesterone contraceptive versus the estrogenic contraceptive. It was a big thing at the time we were studying this. And we got that progesterone activates monominoxidase, and a paper with Harvey Pollard in Pfizer Journal, and estradiol was inhibitory. Some people believe that the estradiol inhibitory action contributes to and its antidepressant activity, which has been shown, by the way, by many group psychiatrists. I don't know, maybe it may have something to do with it. It may have. So that's how we got into progesterone accident at the time at maternity hospital.
Any more questions from maybe from people who are on Zoom?
Who's on Zoom?
No.
Yeah, no?
They are convinced.
So I would like to thank you again, Musa, for coming over and sharing with us first your excitement about the new drug, and second, really congratulate you for a lifelong contribution to the world of Parkinson's neuropharmacology and pathophysiology. Really
Thank you
an
very much.
amazing contribution,
Thank you very much.
and keep on going.